Historically, I'm not very good at this game.
I would say that nucleic acids are my favorite macromolecule to think about, and proteins are probably my favorite macromolecule to talk about. But when it comes to my favorite macromolecule to eat, you can't beat the carbs. You're all undoubtedly familiar with some carbohydrates already, so we've already mentioned starch, and we'll talk about starch a little bit later on. That's a complex carbohydrate, but any sugar is also a carbohydrate. So sucrose is what we call a disaccharide. It's made from two smaller sugars that are linked together, and that's table sugar. That's what people will put in their tea or their coffee if they need it to sweeten up a little bit. And then we have other types of complex carbohydrates we can discuss. We have glycogen, which is a energy storage polysaccharide that is inside your body right now, ready to go in case you run out of blood sugar, if you need some energy real fast. And then we also have things like cellulose. Cellulose is the main structural carbohydrate that supports wood and plant tissues. All plant cells are surrounded by a kind of armor coating. It's called the cell wall, and most of that strength comes from a carbohydrate which is called cellulose, which, unlike all these other carbohydrates, is very, very difficult to digest for animals. In fact, no animal can digest it on their own. They have to outsource that job to certain types of bacteria. So even within something as familiar with carbohydrates, you do get a lot of diversity of function in these molecules. We have the small sugar molecules, which are used for quick energy, glucose, blood sugar, say, long molecules, which are used for energy storage, glycogen, starch, and strong polysaccharides, which are used for structural support, and that would be like cellulose. By the end of this semester, you are going to want to have the chemical formula for glucose memorized. Tattoo it on your chest if you have to. It is C6H12O6, and that in general is the molecular formula for monosaccharides. There will be some number of carbons, in this case 6, twice as many hydrogens, and then the same number of oxygens as you have carbon. So C6H12O6, but you could also make a monosaccharide that's C4H8O4, right? So C to the N, H to the 2N, and O to the N to get that formula. Now, this is the linear structure of glucose, and this is a valid way to write out that formula, where you have the C double bonded to O, this carbonyl group up here at the top. And if you look at the structure of glucose, you can see that it's actually pretty similar to a hydrocarbon. We got hydrogen and carbon in here, and what did we learn about hydrocarbons? Hydrocarbons are high-energy compounds that are hydrophobic on the whole. Well, sugar is just a hydrocarbon with some extra oxygens added in, but oxygen's greedy for electrons, so these are hydrophilic molecules, but they're still high in energy. And one of the places that that energy comes from is this tendency they have to form ring structures. The carbons in a ring are under higher tension than they otherwise would be, and tension means that there's a lot of energy in that system, and that's what we're interested in when we're eating sugars, is getting energy out of them so that our cells can do some work. So when you mix sugar up in water, like glucose up in water, they tend to form these ring structures. Now, to understand this ring structure, take a look at this. In the ring, we have 1, 2, 3, 4, 5 carbons, and then the sixth corner of the ring is an oxygen. What's going on there? Essentially, this carbonyl group, the double bond to oxygen, it's like the bond swung open and bent down and attached to carbon number 5. And then you can see carbon number 6 is hanging off like a little tail on that glucose molecule. So it creates a ring, and oxygen is worked into that ring. In order to simplify the structure of glucose for all future diagrams on these slides, we're going to use this hexagon right here. So whenever you see this green hexagon, it's just a glucose molecule, C6H12O6. Once again, the monomers of carbohydrates are the monosaccharides, the single sugars, the single sweets. Monosaccharides are simple sugars that cannot be broken down by hydrolysis into yet simpler sugars, so you can break them down. You just can't use a hydrolysis reaction and then still get molecules out of that are sugars, right? That's just going to give you some other type of material. So monosaccharides are as simple as it gets and is still considered to be a carbohydrate. Here we have the chemical structure for glucose, should start looking pretty familiar from now, and next to it we have its chemical isomer, fructose. I've mentioned this before, but isomers are chemicals with identical chemical formula, so C6H12O6, but they are arranged structurally different. The carbonyl group is on the terminal end in glucose, and it's one carbon in from the end on fructose. And that slight little change, that one group being moved ever so slightly, is one of the reasons why these sugars behave differently in our systems. Fructose tastes sweeter than glucose does because, for whatever evolutionary reason, our taste buds are programmed to interpret this carbonyl group at that position as being a very, very sweet flavor. On the following slide, we have some additional examples of monosaccharides. So down here, we have our 6-carbon monosaccharides. You guys already know glucose and fructose, but here's another one called galactose. Its chemical formula is also C6H12O6, and this is even more similar to glucose. You can see that this group right here is just flipped around backwards. So you have the same groups attached to the same atoms, it's just the position of this one happens to be a little bit changed. Up here we have the pentoses, so ribose, which is the kind of sugar that you find in RNA molecules. Remember, it's a 5-carbon sugar. Well, all monosaccharides have a similar chemical formula, so if it has 5 carbons, you know that it has 10 hydrogens, and it has 5 oxygens. C to some number, H to twice that number, and O to the first number again. So C to the N, H to the 2N, O to the N. So C5H10O5, and the trioses, monosaccharides that have 3 carbons in their structure, have a similar kind of situation. So C3H6O3. Glyceraldehyde is the only triose that we are going to talk about in this class, and even then, it's only as a product of certain processes in metabolism. You're not going to need to know the structure of glyceraldehyde, but you should know the chemical formula for glucose. All right, well, let's start moving past the monosaccharides, the simple sugars. Let's start building up some larger molecules. Let's get into a disaccharide here. So you all know at this point that a dehydration reaction is what you're going to need in order to put together any 2 monomers. That's what you need in order to put together 2 amino acids, 2 nucleotides, or in this case, 2 monosaccharides. So I'm going to bring glucose and galactose next to each other. They have the requisite OH group, hydroxyl group, and the extra hydrogen right here, and they're going to link together with what we call a glycosidic bond. A glycosidic bond is a bond between a sugar, or molecule derived from a sugar, and the hydroxide group of another molecule, typically another sugar, though it also could be an alcohol or several other things. Hydroxide groups are fairly common. So this is a glycosidic bond between glucose and galactose, and when you have 2 monosaccharides linked together in such a way, we call it a disaccharide, a 2-sugar, a double sweet disaccharide. You're going to be familiar with some of these disaccharides. For example, the one that we just did, glucose and galactose, that is lactose. Lactose is the type of sugar you find in milk, is the type of sugar that the majority of the human race is incapable of digesting because they lack the enzyme to break it down. Next, we have maltose, which you've probably heard of before. Malt beer, malt liquor, malted milk balls, malt milkshakes. That's the kind of sugar you find there, and that is 2 glucose molecules which are linked together through a glycosidic bond. And then you have sucrose, which is table sugar, the normal sugar that you would add in to bake a cake or sweeten up your drink or whatever you're doing with it. And that is the product of a glycosidic bond between a glucose molecule and a fructose molecule. So depending on which monosaccharides are involved, you can get different types of disaccharides, each with slightly different properties. Now, I can see the look on your faces. You're saying monosaccharides, disaccharides, big whoop. I'm here for the big molecules. I want to talk about those complex carbohydrates I hear so much about. Let's talk about polysaccharides. Well, sir, there are 3 polysaccharides that you are going to want to know about for the exam. The first one is called starch, the second one is called glycogen, and the third one is called cellulose. Now, this image of a farmer trying to harvest some potatoes is here to show us where each one of these polysaccharides can be found. So starch is an energy storage polysaccharide. That's its job. It's an energy storage polysaccharide, and you find it inside of plants, particularly very starchy plants. So the potatoes themselves, the cells of the potatoes, contain these little granules of starch. And if we take a look at the structure of starch, you can see that it's glucose linked to a glucose, linked to a glucose, linked to a glucose, 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 all the way down the line. Just a one straight shot of glucose molecules, possibly thousands and thousands of monosaccharides long. That is a starch molecule. Now, starch is pretty good as an energy storage molecule. Whenever a plant needs some extra glucose to break down for energy, it'll just go to its starch reserves and essentially just slice off whatever it needs. Kind of like you might go to a banana and just get yourself a couple of slices for your cereal and then put the rest away. Starch is the same way. We're just going to cleave off a couple of glucoses as needed, put the rest back into storage. So starch is a plant energy storage polysaccharide, but animals also need to store energy, and glucose burns up a little bit too quickly and you need to be able to replace it, and your fat reserves take way too long to metabolize, so we need something that's a little bit in between. We use an energy storage polysaccharide called glycogen, and it will be stored in deposits around some of your muscles, and particularly right around your liver. So that's why it's showing the muscles of this farmer's forearm here, and it's showing you the glycogen. It'll just be in these little deposits all over the body where needed. Now, if we take a look at the structure of glycogen, you'll notice that it is a glucose bonded to a glucose, bonded to a glucose, 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 all the way down. But unlike the starch, it's a branched molecule. You actually have multiple strings of glucose going off in many different directions, and that branching makes this overall a much more efficient storage mechanism for energy. You're able to pack more energy, more glucose into a much smaller space using glycogen than you would for starch. So starch is a plant energy storage polysaccharide, but glycogen is an animal energy storage polysaccharide. Once you're going for a run and you run through all of your blood sugar, the next place your body's going to look for energy is your glycogen reserve. So it'll start converting that glycogen over to glucose, and then only once you're out of all of that will it start getting around to metabolizing your fat. One more polysaccharide to know about, and it is called cellulose. Cellulose is the most abundant organic compound on Earth. Okay, if we were to weigh up all of the different chemicals that we talk about this year, all the different organic materials, cellulose would be the one with the largest amount of mass. It is not an energy storage polysaccharide. It's actually very, very difficult to digest. Even herbivores, like animals that eat plants like leaves for their nutrition, do not make the correct enzyme to be able to break down the bonds between the sugar molecules inside of cellulose. They have to have bacteria that symbiotically live in their gut, and those bacteria, some of them can produce that enzyme, but no animal can actually do it entirely by themselves. So there must be some really crazy monosaccharides in there to give it that much strength and resilience. So let's take a look at its structure. Glucose, 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 glucose. This looks just like a starch molecule, just a bunch of them laid side by side. But there is a little bit of a difference here. If you take a look up at starch, it's a straight line of glucoses going down the line, and take a look at the oxygen in the glycosidic bond. It's pointing down in every single case. Down, 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 down. If you take a look at these cellulose glucoses, one's pointing down, the next one's pointing up. One's pointing down, next one's pointing up, down, up, down, up, down, up, down, all the way down the line, and same thing with the next string. As it turns out, that bond, it's called a beta-glycosidic bond, is very difficult to break apart for animals. And what effectively this means is that every glucose has been flipped upside down relative to the other ones. Every other one in the chain is oriented the other way around. So parallel strands of cellulose can hydrogen bond with each other. And what do we know about hydrogen bonds? Weak on their own, but if you have very, very long stretches of hydrogen bonds, that force can add up and actually be worth quite a lot. So these threads of cellulose, these individual strings, they hydrogen bond next to each other and they form fibrils of cellulose, kind of like ropes of cellulose, which then are arranged in a kind of woven patchwork. And it's that woven structure of cellulose fibrils that gives plant cells, plant cell walls, an organelle that we're going to talk about in the next unit, how all of their structural strength, and it's what gives wood a lot of its strength as well. There's other materials in there as well, like lignin, but to give you an analogy, if you've ever been to a construction site and they're tearing down, I guess it would be a destruction site, they're tearing down a building so there's rubble all over the place, you look at some of those concrete blocks that they've torn apart and you see these iron bars sticking out of them, reinforcement bar, rebar it's called, so they have reinforced concrete there. What we do to reinforce concrete is we run iron bars going every which way inside in order to give it a nice sturdy framework to work with. Cellulose fibrils are arranged in exactly the same way. Just like reinforced concrete gets extra strength and extra support from the iron bars that are worked into its structure, the cellulose can also build up the strength of plants and plant tissues and plant cell walls by having this nice woven pattern. So starch and glycogen are energy storage polysaccharides, one's for plants, the other for animals. Cellulose is a structural polysaccharide. It's there in order to create strength. There are other structural polysaccharides. We won't talk about them too much, but for example, in the animal kingdom, there is a polysaccharide called chitin, and it's the kind of thing that you would find inside of insect shells or like lobster shells, that same kind of material there. And it's also what the cell walls of fungi, funguses, mushrooms, and that sort of thing are made out of as well. So there are other polysaccharides, but the three big ones are going to be starch, glycogen, and cellulose for this unit.
Oh, ooh. I can just keep making more bagels. This is the escapism of video games right here. This is the world I want to live in. I don't care that I'm getting a terrible time